My name is George Portugal, and I am a senior application scientist at Harvard Bioscience. And today, what we're going to be doing is having a discussion about toxicology research and how Harvard Bioscience provides a holistic approach for addressing questions related to toxicology. Okay, so as it should be clear to anyone doing tox research, trying to understand toxicological effects can be very complex. So you have a number of different factors that can uh, generate a toxicological effect, whether it's neurotoxicology or cardiotoxicity. And when you're putting together a publication for doing neurotox or cardiotox, it's very helpful if you can integrate multiple techniques in order to understand that effect. The reason being that if you have 4 or 5 different techniques that are all pointing in the same direction, it suggests that you have something really powerful there. It helps to increase the impact of your manuscript and get it into a higher impact journal. And it's also helpful if you can validate discoveries, both going from a cellular model where you're looking at, say, a neuronal or cardiac cell culture, and then trying to validate that in a whole animal model, or perhaps in an organoid, or going in the other direction where you discover toxicological effects in a whole animal model, and then you go into a cellular and molecular direction with cell cultures and try to understand the mechanisms that underlie it. Having integration of multiple techniques and doing in vivo and in vitro modeling all at once can really strengthen the conclusions of a publication. All of that being said, though, it's very difficult to accomplish this. So I have this illustration here of a patch clamp rig with a bunch of different imaging approaches at once. Kind of to illustrate this, it's hard to take hardware and software from 7 or 8 different companies, put them all together, and hope that they work. A lot of times there are various incompatibilities. You may have to end up returning products or finding new products or substitutions in order to get things to work. Another challenge that comes with integration is installing the instruments and then training staff with these new techniques. So with most labs, the PI might have a certain skill set, a certain number of techniques that they're proficient at, but they have limited time to work in the lab themselves. They're focused on generating grants, editing manuscripts, mostly working on the written component of a lab. And so taking time to train new people can be difficult. And even if there are members within a lab that are proficient at a specific technique, their time in the lab is usually limited. And so if they don't transfer that information over to new members, then it can easily get lost. And then finally, data analysis can also be a challenge because even if you're very proficient at collecting data from a new technique that you've added, you may not necessarily have the experience with interpreting it or looking at it in a variety of different methods to kind of get the most out of the data that you've collected. And so Harvard Bioscience really does, our core mission really is to have this integrative lab approach. So we are a collection of a number of different companies. So Harvard Bioscience is the parent company, and we have a number of different companies that are part of that larger conglomerate. So we have HICA and Multi-Channel Systems and Warner Instruments for electrophysiology. We have PanLab for behavior. We have DSI for telemetry. And we have BTX for electroporation. And that's just a few examples of the companies that are part of Harvard Bioscience. So collectively, what we do is we focus on integrating these various techniques and trying to reduce incompatibilities between them. We have a staff of scientists such as myself who are trained not only in neuroscience, but also cardiovascular, metabolism, respiratory, and we share that expertise with our clients. We work together to bridge these gaps in these multiple system investigations. The other advantage of working with us is that we support all stages of research. So we have folks like myself who are very proficient at cellular and molecular neuroscience or cardiology, and so we can help with assisting with those kinds of discoveries. And then we have others within the organization that are very proficient with in vivo techniques. And so if you're looking at trying to take discoveries from a cellular molecular perspective and then validating those in a whole animal model. We have people who can help with that, and we have people who can assist in the other direction as well. So for the remainder of the talk, what we're going to be doing is discussing a number of examples of publications that have used our instruments for cardiotox research, for neurotox research, and finally a little bit at the end about inhalation as well. So to start, we'll be beginning with cardiac toxicity, and the first thing that I'll be discussing is the HICA patch clamp amplifiers that we provide. So the HICA amplifiers are the gold standard for patch clamp electrophysiology. It's a fully digital system which has the amplifier and the digitizer all integrated into a single unit, and it connects very easily up to a computer through USB, and it also can be integrated with a variety of other techniques, including imaging. So I have two examples here of using a HECA amplifier for cardiac toxicity. So Karnik and colleagues were specifically looking at hERG channels, and they have it's pretty well established that endocytosis of hERG can lead to QT prolongation and increase of arrhythmia. And so with this particular group, they were looking at ARF6, which can increase hERG channel density. And so this is shown here in the top panel using the HICA patch clamp amplifiers. So it shows that it's very important for that endocytosis. Shaper Welling and colleagues also used the HICA patch clamp amplifier to look at NAV 1.5. So variations in NAV 1.5 are associated with risks for arrhythmia. And so very specifically, this group was looking at a point mutation of NAV 1.5. It's the S106G mutation. And it was shown that cells expressing this variant have a gain of function when compared to other variants. And so that's what you can see here on the bottom panels. So Harvard Bioscience also has microelectrode array technology for use with cardiac toxicity. So this comes from a company known as Multi-Channel Systems. So for those of you that don't know, a microelectrode array is a network of 60 or more extracellular electrodes that are normally plated on glass. And so the advantage of this technique for cardiac research is that you can simultaneously record from all channels at once. And so that means that you can get information using a cell culture of where a pacemaker is located, and you can monitor things like changes in conduction velocity, QT prolongation, field potential duration. And we have a variety of different designs for this technology as well. So on the right over here, you can see that we have a multi-well plate. So this allows you to do high throughput recordings using cardiac cell cultures. So this one particular study by Ma and colleagues is looking at cardiomyocytes, and so you can see that there are changes in conduction velocity in those top panels using different compounds. So isoproterenol, for example, has an effect on the conduction velocity, and iso with PFD also has an effect as well. And I also have this video here just to illustrate multi-well MEA recordings using human iPSC-derived cardiomyocytes. And so you can see on the left-hand side, it's a 24-well plate. Most of the wells have cell cultures on them, and so you can see the conduction velocity from each one. You can also, again, monitor changes in QT, look at changes in T wave. And so it's a very powerful tool for looking at toxicological effects on cardiomyocytes. Recently, we have also partnered with another company known as 4C Biosystems, and they have designed a product that pairs really perfectly with our microelectrode array recording systems, and it does what's known as laser optoporation. So what does that mean? So normally when you are recording from cardiomyocytes on an MEA, you're getting field potentials. You're getting extracellular recordings from those cardiomyocytes. Now, what the 4C instrument does is it has a laser that can be very precisely positioned underneath all of the electrodes on an MEA. And once that laser is fired, it optoporates the cells, and then the waveforms go from a field potential to an action potential. And that's actually what you can see in this image right here. So what's really nice about this design is that instead of looking at action potentials using other techniques like patch clamp electrophysiology, which can be slower and lower throughput, this is a much higher throughput kind of a technique. It's also very long lasting and it's repeatable up to a month. And so then you can look at the effects of compounds on AP, including toxicological effects, using the 4C Biosystems instrument paired with our MEA recording systems. And then finally, we also have techniques to do microelectrode array recordings with isolated heart preparations. So what you can see here is an example of our Hugo Sachs Langendorf heart preparation. So you can have an isolated heart, which is kept alive and beating, and then you can use our flexible microelectrode arrays, and they can be positioned directly onto the atria or ventricles, and you can monitor the same types of metrics that we have discussed earlier. So specifically, you can look at changes in QT prolongation, field potential duration, and conduction velocity. Now, moving on to an in vivo approach, we also have telemetry devices from DSI, and these can be implanted into animals, and you can do continuous monitoring of a variety of metrics that are important for cardiac toxicity, including changes in blood pressure, both systolic and diastolic, changes in mean arterial blood pressure, and also changes in heart rate. So an example that I have here is by Carlson and colleagues. And so this group was looking at the effects of 3 different anesthesia protocols on heart rate. They were using an animal model. These are Göttingen mini pigs. So these are a type of pig that are used for cardiac research, and they're really an ideal animal model because their cardiovascular circuitry is very similar to that with what you see in humans. So using these DSI telemeters, they could monitor the effects of blood pressure and mean arterial blood pressure and heart rate over a very extended period of time of 10 weeks. And what's also nice about this technique is that there's minimal interaction required by the investigator as well. Okay, so moving on to neurotoxicity, we'll be discussing a little bit about the HECA patch clamp amplifier because, again, as you can imagine, there are hundreds of studies using this technology for neurotox research. So one example that I have here is by Loser and colleagues, and they were also examining the NAV 1.1 and 1.2 channels. Specifically, they were looking at the effects of TTX and lidocaine on NAV function. And what they found is that you could do use-dependent blockade of NAV. So multiple pulses of lidocaine lead to a faster blockade, whereas this kind of effect was not present with TTX. Okay, so microelectrode array recording technology has also been very widely used for neurotox research, specifically looking at epileptic events. And so it's very easy to use an MEA recording system to monitor changes in epileptic activity on a brain slice, on iPSC-derived neuronal cultures, on standard monolayers, and also with organoids. And so the example that I have here was done by Hedrick and colleagues, and they were looking at mutations in NAV 1.1 and seeing if that had a risk for epilepsy. So in this case, there are mice that are carrying a human mutation of NAV 1.1, and you can see the activity from those mice on the bottom panel. And as you can see with the spike raster plots, there's a very clear difference between the wild types and the mice that are carrying this mutation. So they have very pronounced synchronized bursting, extremely active, and then followed by periods of relative inactivity, whereas with the wild types, that's really not something that you're seeing. And so, yeah, it's a very powerful technique for looking at epileptic events, and you can also monitor these things in real time. So this is an example that I recorded in mouse. These are hippocampal brain slices. And so what you can see here is real-time monitoring of epileptic events in low magnesium. And so, yeah, you can see here the epileptic event has started. And so not only can you see where it starts, but you can see how it propagates across the entire hippocampus. And that data is also being overlaid here with the imaging that was being done in real time. So DSI telemetry is also very widely used for monitoring epileptic events in whole animal studies. So an example that I have here is by Andrew and colleagues, and they were looking at organophosphorus-induced seizures. And so uh, what they were specifically looking at is whether alpha-4 containing nicotinic acetylcholine receptors have an effect on these seizures. And so you can see actually here on panel B that they're applying some antagonists that are specific to those alpha-4 containing nicotinic acetylcholine receptors. So DH-beta E and mecamelamine, those are both selective for those alpha-4 containing nicotinic receptors. And so you can see that epileptic events are blocked in those animals, whereas in some of the other examples, if they're exposed to the organophosphorus, they do have seizures. And again, this can be done over extensive periods of time as well. I have another example here by Laporte and colleagues. And in this particular study, they're actually using both our MEA recording systems and our DSI telemetry to study epilepsy. So this is a really nice example of doing both a whole animal and a cellular model in the same study and arriving at very similar conclusions, which is rather powerful. So in this slide here, you can see that they're looking at ALEX conditional knockout mice. So ALEX is a protein that's involved in synaptic endocytosis during seizure events. So the animals that are missing ALEX, they show quite different effects on the MEA. And then when moving to a whole animal model and looking at telemetry, you also see fewer seizure events in the conditional knockout mice. So again, both of the MEA data and the DSI telemetry data support each other. So the benefit is that you can easily validate results from an in vitro model here in a whole animal or vice versa. It's also worth discussing one of the newer products that we have just released, which can be used for looking at neurotox effects, and that is the Viva Mars mobile activity rack system. And so the advantage to using this instrument is that you can do high throughput drug screening, neurotoxicity, and neuropharmacology. It works primarily with rats and mice. It uses a high resolution infrared beam technology to monitor activity. And the advantage also is that it's being run by Panema software, which adds GLP compliance. And then finally, I'll briefly be discussing some of the solutions that we have for monitoring inhalation and how they relate specifically to toxicology. So one of the challenges of doing inhalation research is that understanding what kind of dose is being administered to the subjects of the study it is very difficult. And in a lot of examples, people do estimations of exposure rather than something precise. Whereas with the Buxco inhalation towers that we provide, you can get very precise aerosol delivery to each individual animal, despite their differences in their breathing rates and other manipulations that you might have done to those animals. So you also have an understanding of the exact dosages that are delivered in real time, and that's also a unique feature that isn't present with other methods of monitoring inhalation. The other thing that's very nice is that it's fairly automated. You can control the entire inhalation tower through software and understand exactly how the aerosols are moving, stop them when you need to stop them, and you have a very precise dose. So to that end, very recently, we had a collaboration with Merck looking at our inhalation tower and some of the compounds that they're currently working on. This data was recently presented at the Society for Toxicology Research, and we expect, publications forthcoming and other collaborations with them as well. So to conclude, Harvard Bioscience really does provide a full portfolio of products that can help you to meet your research objectives and aid in more scientific discovery, whether that's in vivo or in vitro, whether it's something you're well versed in or something that's totally new to you. Our instruments from our different product lines work together. They don't work in a vacuum. So they combine together, and they do so easily, and that can help you to have more powerful, impactful discoveries. You also can be assured that when you buy instruments from us that you're not working alone. We have a team of talented scientific investigators who can work with you and help you at all stages of your research, whether it's something brand new or something you've been doing for years. Um, the fact that our instruments interact also allows for more dynamic and complex experiments. And then finally, the fact that we have instruments for in vivo and in vitro research allows you to do validation kinds of work.